Maybe maybe they vanished into that realm apart where all missing items go.
No, they're in the basement.
I don't think of where I saw them.
Okay, I know that chances are I'm preaching to the choir here, but I may have someone in here who is on my list of students who didn't take the first lab quest that was due last night.
That's my class.
Can someone mute their mic?
It was just stomach pain.
Yeah, nothing else in it.
Can someone mute their mic?
Okay. He was like running back and forth to the bathroom, so. Okay, diarrhea.
Let's see if I can mute them. Okay, so I might be preaching to the choir here in that I'm not sharing my screen with you yet because I don't want you to see people's grades, but I can see that I have at least 2 people here. I have at least 3 people in attendance right now who didn't do the first lab quest that was due last night. If you know, that's 40 points. And so what that means, and I'm going to do the math really quick here. What that means is that your grade has already dropped 3%. And the reason I'm telling you this is because partly to get you motivated, because I don't know how else to get you motivated to get your assignments in, get your lab exercises in, get your chapter quizzes done. I know that we have not finished chapter 1. And it looks like everyone got the first chapter 1 quiz done, but you should all have access to the lab manual at this point. Nobody has told me that they don't have access to it now. And I'm saying this to get you motivated because midterms are due in less than a month. And if you aren't getting the lab quests done and you aren't getting the assignments turned in, you're technically failing. So the due dates, all the due dates are right. Bear with me here.
I just wanted to share with people. I'm sorry.
No, give me 1 second, Lakeisha, okay?
Okay.
If you come, I'm sharing my screen with you now. If you come right here, especially for McGraw-Hill, all of your due dates are right here. Most of this, I wouldn't put it off until October 4th or September 30th. I would get it done now because this is a lot to try to do the evening of October 4th. We've already done chapter 1, so we're not going to go back to it. The virtual lab, you're in a class now, and you have to stay on top of the work or you're going to fall behind. And I'm saying this, and the reason I'm saying this is partly, I'm mainly saying it because I don't like failing students. There's very few things in my life that I like least. I don't like failing students. But if you don't do the work in the class, I won't pass you either. And so please get going with the work. If you can't access it, you need to let me know. And if it's not loading, the first thing that you need to do is call, if it's McGraw-Hill, call McGraw-Hill. Because chances are you have a pop-up blocker that's disabling you from being able to open something and get something to load.
So that's why I wanted to mention, Mary, that it's not a pop-up blocker all the time. For people that don't know, you need to download a different browser because that's what I had to do. I was using Chrome and I download Firefox to get access to the labs.
You know, and sometimes it depends on what your computer, because I honestly don't use anything but Chrome, but I always have it load on Chrome. And so she's right, you may need to try a different browser, depending on your computer, but I would still contact them. Because I know there is a student here, and I think she's here, that between she and we put in 4 calls to Wiley about getting her into the lab and allowing her to access a lab. And I finally got home and called them last night, and I can say that they're probably not my favorite company at this point, because I feel like some people are always on a learning curve there. And when I call, they may not be able to, you know, we had 3, she called, she knows who she is. She called one time on Thursday, not Thursday, Tuesday. I called twice Tuesday night. Then I called yesterday, and they were telling me different things than what they were telling me Tuesday night. And I got a little cranky with them. Because they wanted us to all be she and I could all be in the same room. And I said, you realize this is a remote class and we may never meet? And then she started looking into it more and she helped me solve the problem. And so I'm more than willing to make calls and I'm more than willing to figure out why you can't make it work. And like Lakeisha said, if it's your computer, a lot of times I can't troubleshoot that. I don't know enough about computers to troubleshoot someone else's computer. That isn't my expertise. But we do have a hack IT tech department, and they are really good. And there is a Wiley IT tech department, and there is a McGraw-Hill IT tech department. And I can see, at least with hack and McGraw-Hill, I've always gotten positive results. With Wiley, sometimes I have to spend a lot more time on the phone to get in the same area, but I still get the problem solved. You have to do the work. If you don't do the work, you will fail the course.
Labs 1, 2, 3, and 4 are still open for a later date, right?
They are, but I think I've got people who are putting them off and putting them off.
All right, sorry.
Yeah, so the reason these are open, these practices are open until the 4th is because that's when your first exam opens. And so I want you to be able to use it as a study tool. Does that mean that you shouldn't start using them until right before the exam? No. Because you realize that there, as you've probably figured out, there's a lot of material in this course. Start studying it now.
Where did you say we found the chapter 2 practice quizzes at again?
They are they're under the week. So if you come in,
Okay.
the due dates are right there. But if you come in,
it's not due yet, is it? I can't see it on the
it's
screen.
not due yet because none of the practices are going to be due until the day of the exam, because I want you to be able to use them as a study tool. If you want to know if you want to know what you need to know and what you don't know yet that you need to know for the exam, do those chapter practices, and you can do them repeatedly.
Okay, thank you.
You're welcome. But here's the chapter 3 practice right here. Here's the chapter 2 practice. Here's the chapter 1 practice.
Now, Mary, do you want the chapter 3 worksheets? Are those just for like us, the students?
Those are just for you.
Okay, thank you.
You're welcome.
So I was freaking out over handing them in.
No, don't hand them in.
Would we have a class like to dedicate maybe to going through that?
To going through what?
Through the worksheets?
You can ask me questions. You can ask me questions after class, but I can't dedicate a class to it right now.
Okay.
I also would not be opposed to maybe like getting the student email list and seeing maybe if some people want to form like a group Zoom study group.
That's probably a really, really great idea. I'm trying to think how you would do that. I just recently got, I think someone is going to the tutoring center here at HAC or contacting them or coordinating with them. Because I just recently got an email from the tutoring coordinator at HAC. And I can tell you that in the last year that I've worked with HAC, this is the first time I've ever received an email from her. So there is there
we
is,
should-
go
we
ahead.
should- I said, I think that's a good idea that we could do that and like go over things with each other. If we don't have time to do class, we should meet up after.
How,
Yeah, because the information is coming like very quickly. Like quickly.
All of us under one thing.
We could all in the chat, probably just to clear this up with the professor, we could all just probably put our emails in the chat, and then somebody can, either me or whoever I'm speaking to right now, we could get like a mass email sent out and be like, hey, we can make a Zoom link. You want to meet on Fridays? Do you want to meet on this day? Do you want to meet on that day? What days work for every other student?
I'm
You're
pretty sure you
talking-
can look at the
Could
class
we
list.
talk through here, through the discussion? Like, could you open up a thread that we could all?
I don't know how to do that. I'm trying to think. How would I do that?
I mean, if you look in the chat, I'll put my email in the
Discussion
chat.
list. Create a new forum, a new topic. Let's see if I create a new topic. Let's see what it does. Group, everyone can access this
Like-
topic.
This thread could just be there forever. Like, whereas if we talk through chat right now, it's gone by the end.
Okay, well,
that's true.
let's. So, do you see what I'm, you see my screen here?
Yeah.
New forum exists. New forum, let's see what this does. Study group. Let's see what it does. Okay, title. Email addresses needed. For those for students wanting to study as a group. Join a study as a group. Yeah, that's a good word. Study as a group. Allow anonymous posts. Users must start a thread. No ratings. Now, someone else try to open it and tell me what it does.
It lets you start a new thread.
So does it allow you to add an email? Or add a post?
Let me see. What is our domain for our Hawkmail? Is it at hack.edu?
I don't know. It might, I think it's Hotmail from what I understand for you guys. I don't have a.
At
Yeah.
hotmail.hack.edu.
.edu.
Okay, I posted something.
Okay, yeah, I can see it.
You can see it. I can't see it, but you can see it.
Yeah.
View topic. Let me just.
Are we doing like our regular emails then?
Oh, no, I see Miranda's. Yes.
I
look
put
here.
I put both. I put my regular and my Hawkmail because I have my Gmail accounts linked up together. So either way, I'll get it.
Yeah. We should be able to like go, like I can answer you, like I can talk under yours, you know?
You can reply right here, I think.
Where's the reply?
Let's see here.
Well, I can just,
Can you
So
reply?
Professor, if you were to click on her name at the top,
Yeah,
if you click on that, it'll bring you to the actual post and then
Reply
you
to
can
thread.
reply to it.
okay.
oh, okay. I got it.
I'll put my email in there if you have any questions about D2L. Honestly, I've used it before, so I kind of know the ins and outs of it.
Awesome,
So I'll put my email in there, and if anyone has any questions, you can just email me
awesome.
about D2L specifically.
Well, Genesis, you probably know more about it than I do. Okay. Other questions or other issues?
So I just want to clarify, Professor, because I caught like the end of what you were saying. I was excusing myself to the bathroom. With our due dates in the calendar, that is only our labs, correct?
I think that's also the practice. That's mainly the practices and the chapter quizzes and the smart books. The labs they are due every time we do a lab, like we're going to do a lab today. Every time we do a lab, the link is going to be posted under that same week. This stuff, bear with me, Miranda, I'm sorry. Let me go back to answering your question. So this is just for stuff from McGraw-Hill.
Okay. So there are other assignments due, because that's where I'm struggling. I'm hitting the due dates. Why am I still missing content? And I'm not realizing I have to go in the chapters of the week and follow that list as well.
The only thing that isn't listed here. Yeah, okay, so the only thing that isn't listed here are the lab assignments from Wiley.
Okay. So
And
McGraw-Hill is the calendar, Wiley is in the content.
yeah,
Thank you. I've just been really struggling with the due dates because I've been following the calendar, and the calendar, I'm like, I'm missing things.
yeah.
And I just been stressing out about due dates.
Why McGraw-Hill is integrated that way and Wiley isn't, I have no idea.
Okay.
I wish I knew,
Thank you for clarifying.
but yeah. Other questions, other issues?
So I have 2 of my labs that I did, but are saying that I didn't do them.
Can you
So-
send me, Genesis, can you send me a screenshot showing the name of the assignment and the score that you received?
Yeah, so that's the thing. In D2L, it's saying that I didn't do them, but also when I think it's Wiley, it's also saying I didn't do them. So
Well-
if I were to just redo those, would I be able to get like
Yes,
full
but
credit?
one other thing that there may be that you're not getting is up, I think up in the right-hand corner, when you're doing one of the, actually, let's do this. Someone share their screen with me. Bear with me just for a second here. Someone besides me share their screen. And
Yeah,
go
mine's
into-
not either. Like for 1 and 2, labs 1 and 2, I believe it was. It's saying it's not submitted. But I think in Wiley it's still showing I did it.
Well, go in, do me a favor, someone share their screen and go into this assignment right here.
Anybody
I'm
share?
sharing my screen right now. Where would you like me to go?
So click on this assignment right here under this week.
I don't think you see my screen because I'm sharing my
I
screen.
I
can't
don't see it. Well, and now
You
I
have to stop
now I
sharing.
should see it.
Let me
Okay,
stop
I see it.
sharing.
I see, nope, go back. I saw it for a second and I was doing something wrong.
Okay.
Okay, go down to, scroll down. Go down week 9, 2021, click on that. Now go down, scroll down, click on that. Click on lab exercise 6. Now click on the first, that one, click on that. Now go down, open assignment. Start assignment. and then put them in place. I'll tell you what the answers are here. Cytoplasm goes in, you can't see my screen. Cytoplasm goes in the third one down. Actually, no, it doesn't. I'm sorry. Cytoplasm, put in the third one down, put fat storage area. Nucleus goes in the fourth one down. Blood vessel goes in, I'm making sure here. Put blood vessel in number 5. Let's see what that does. Put in the top one, put plasma membrane. Let's see what it does. Click submit. Okay, it was partially correct. So flip those two around. Okay, now scroll down. Okay, go to the next question. What I'm curious with is at the very end of your assignment, if your assignments are not showing up, is
Like
there
somewhere
a submit
here.
box?
It's,
And-
yeah, there's a submit box somewhere in like this corner up here. I was losing it too. Like I was not catching that submit box either.
Some
Yeah.
of them might have a submit box and some of them don't though.
Yeah,
Okay, so it's not just me.
mine says you have a submit box for lab 5.
No,
Say it
one of them I
again?
did twice and it's still not saying submitted in the submit box.
Does it, the person who says they submitted it twice, but it's not giving you a score, did you get a score? Did it say this is what you got, you just can't submit it?
Yeah, it said I got like an 8. I think I got an 80 on it.
Okay, so what I need you to do is send me a screenshot with that information. And it doesn't matter what happens with Wiley, I'll still put that grade in.
Okay, because I'm dumb and I delete emails. Like it came through the email, I think. And I like delete them because I'm like
Can you go,
OCD and
can
like
you go, did you clear it completely or can you go to the deleted file and find it?
I probably deleted it. I mean, if I go back into the lab, would it say I completed it or something?
Yes, it will, Ashley. Yes, it will. It will have your score there.
Okay, it's just lab exercises 1 and 2, I think. So let me. The AMP, where is it?
So I'll do that too. Then I'll screenshot mine.
Okay,
Yeah,
Yeah,
if I can get in there, I'll screenshot it.
if you screenshot it, then I can update your grade regardless. But I know, and
Okay.
I don't know if I think on the McGraw-Hill, as far as I know, there's always a submit option. I don't know if it's on Wiley or not. Wiley is a new text publisher for me this semester to work with. But I do know that it's on McGraw-Hill, and I know that's probably one of the main reasons people are not getting their scores posted for McGraw-Hill assignments. Other questions, other issues? So eventually, within the
Professor,
next,
one more. Sorry.
Wow.
That test I just did or that lab I just did, it's saying
Yeah.
I have a 0.15 out of 5. Am I gonna be able to go back and fix that?
yes.
Since I exited
You
the
have
lab.
unlimited attempts.
Okay,
Miranda.
okay. Just wanted to make sure. Thank you.
Yeah, of course. If it doesn't let you do that, let me know. Because I think after our adventures, Miranda's a student that we made lots of calls with over the last couple of days. After our adventures, I may be slowly learning how to fix that. So let me know if it doesn't work. Cece.
Okay, so I just got into my lab exercises 1 and 2, and
Uh-huh.
it's saying I did all 100 questions, which is weird because it says I got a 0.05 out of 0.05 time spent. But saying I spent 27 seconds, but how that makes no sense. Like,
Actually, is it giving you a score at all besides a 0.0 something or other?
it's a score on the top right corner, or the grade, like the final grade. Because it's saying something. It is giving me a score at the top.
What one was it? Was it the lab exercise 1 and 2?
Yeah,
Okay, give me 1 second.
a bunch of questions, and I think I did that like once or twice.
Give me one
and I
moment.
completed it.
Let
You're
me see.
good.
Let me see if I can figure it out. I have to remember what they told me last night.
Yeah.
Added due date.
It says 1 hour and 7 minutes.
Is this
Maybe that's-
Ashley? Ashley, is this
Yeah.
Ashley? Okay, give me one
Yeah.
second.
Unless that was just like the total time we had. I don't know. I have no idea. I definitely did that one.
Ashley, see if you can't get into it now.
Okay. It's just saying continue assignment, so that's what I'm doing.
Okay,
And
so
it's
just
saying-
continue the assignment. Have you done it before? Is that what you're
Yeah,
saying?
I made it. That's what I'm saying. I did 100 questions. It's saying I'm right now. I went to continue assignment. It's saying 23 out of 100 questions.
Okay, give me, let me look for a minute longer, all right? I'm
Okay,
still learning Wiley a lot.
so that's the one that took like forever. I remember
Okay.
because I had like 100 questions.
It's saying that you got a 74%. Does that sound about right?
Yeah, I think so. I got like an 80 or 74. Maybe it wasn't 74. Is that the final grade? It's
Okay.
my final grade.
Well,
now?
if you got a 74, you're gonna get an 8. Don't redo it. Give me 1 second here.
Okay.
I'm not sharing my screen with you, right? You don't see my screen.
Let's see. Sorry.
Do you-
Yeah, now
do
I
you-
do. No, you're
do
not
you-
sharing your screen.
Okay. I don't want you to see my screen for a second here.
Okay.
You know what? Give me 1 moment here.
Okay. Yeah, it was just
Mom-
frustrating because I know that one took forever.
Mom? Yeah. Would you grab my notebook out of my bag? Out of where? Out of my bag here. And I think Leo is sitting on a pen. Yep. There's a pen right there. Just grab me that spiral notebook. Yeah. Okay, so for everyone, Jamie, I have you too. There are 4 people who have not started it according to this.
They can see you. I was just not listening. Love you, bye.
And just give me 1 more moment. I'm sorry. I apologize. I'm just going to mention some names. And if it's a FERPA violation and you get mad, feel free to let me know afterwards.
I'm not gonna be mad.
Okay. Aaron, you got 100%. Miranda, you got 100%. This is for lab exercises 1 and 2A. Jamie, you got 100%. Jesse, you got 100%. Rachel, you got 100%. Jessica, you got 100%. Where's Jessica's? I don't see it on here. Jessica, 100%. Lakeisha, Lakeisha is showing a 0 score for you. Catherine,
It's showing what?
Lakeisha, it's showing you got a 0 so far.
Because I was not able to get access to the labs. I emailed you about it, remember? So you had left it open for extended.
Do you have access to it now?
Yes, I have access to it now. That's why I did lab 5, but it's still open, right? Because we was having trouble in the beginning.
Go and try to get into lab exercises after lab. Try to get into lab exercises 1 and 2, and tell me what it does.
I don't like my phone being set in front of people.
I understand. Isabel, you got 100. Terry, you got 100. Hannah, you got 100. Bianca, you got 100. Yogesh, you got 100. Stacy, you got a little bit. Asante, you got 100, and Michaela Costley, you got 100. And I'm just saying that because I want you to know if your name wasn't mentioned there, I don't have a score for you.
What are you talking about? What score for what? I'm sorry.
Lab exercises 1 and 2A.
Did you call my name?
I don't know because I don't know who's talking to me.
Me.
Who's me?
Catherine.
Sorry. I'm trying to remember if I did or if I didn't. Yeah, I did. You got 100.
Oh, okay. You scared me.
Professor, I'm also noticing in the grades that there are 3 parts that say lab exercises 1 and 2. So like one says like dash out of 10, and then I have 1 and 2, 5 out of 5, and then 1 and 2B, 5 out of 5. So I don't know if that's
there's one that says 10.
Yeah, there's one that says a dash and then slash and then 10.
Let me look.
And it's above chapter 1 quiz, and below chapter 1 quiz is lab exercises 1 and 2, and then it says lab exercises 1 and 2B.
All right, because I didn't put in that 1 and 2A there. That's A.
Because there are 3 of them though total.
No, oh, you're right. Shoot. Let's see here. Bear with me. Dropbox. We're going to delete this one. More actions. It's still there. Give me 1 second here. Edit. You should not be there. I just deleted it, but why is it not letting me delete the Dropbox? Let's try something else. Save and close. Oh, because I'm not gonna. You're right, there is. I don't know why that's there. Let's just do this. I'm
Okay,
just
so
kidding.
we can take we can take the chapter 2 quiz and all the like the chapter 2 practices. It's not chapter 3, right? We can do that now.
Yep, you can.
Okay. And pretty much everything on the calendar except for the newer quizzes, right? Like chapter 3
Right.
quiz. Like don't take those yet. Okay.
Yeah. I'm just for this lab exercises 1 and 2 that's giving you 3. That's your third one that I can't manage to get rid of it on the grade box. We're just going to do this. Enter grades. I'm just going to give them as extra credit 10, 10, everyone's getting 10 points for it. I can't get rid of it, so 10, 10. 10. So for that third column, that was 10 points for lab exercises 1 and 2. I just gave everyone 10 points. Because I can't manage to get rid of it, so we're just gonna do it that way.
Well, thank you anyways.
You're welcome. You know what? I'm gonna do one more thing. Oh yeah, I'll worry about it later. Other issues? Okay, I think also I finally figured out after our class on Tuesday, how do I say this? How to allow the screen to move along smoothly and not zoom in and zoom out for no reason whatsoever. So we shouldn't have that problem today.
The lab quest, we're only able to do it twice, or is it
The lab
unlimited?
quest, yes, the lab quest, because it's bigger than your typical quiz. You can only do it twice.
Okay.
I'll extend out the deadline on it this time. But I won't do it in the future because the deadlines are also on the syllabus for that.
Okay.
Be patient with me for 1 second. I'm just extending the deadline here. I'll extend it out another week. So it will be due on the 29th at midnight.
What assignment is that?
That's the lab quest. That's the first lab quest
Okay.
on McGraw-Hill.
The first
Okay.
one?
Yeah, because I had a few people who didn't get it done.
Okay.
And especially because it's the first one, let me just extend out the deadline on it. All right, now I share my screen. So today we're going to talk about 2 different tissues. We're going to talk about epithelial tissue and connective tissue. Next week's lab is going to be fairly easy. This week's lab is going to be more involved. So you've been warned. There's 4 basic tissue types that make up the human body. We have epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Epithelial tissue is also used, it's involved in gland formation, and it lines all body surfaces, so it's all body membranes. If you remember with the first day of lab, we talked about visceral and parietal membranes. Visceral membranes meaning the membrane that's right on top of the surface of an organ. That's epithelial tissue. If you think tissue that lines cavities, that's epithelial tissue. So the body's composed of trillions of cells, which are organized into more complex units called tissues. If you remember on that hierarchy of complexity, we went from atoms to molecules, to the first chapter, atoms to molecules to macromolecules to cells, no, to organelles, and then to cells, and then to tissues. So a tissue is a group of similar cells and extracellular products that perform a common function. Four basic types: epithelial, connective, muscle, and nervous tissue. Tissues vary in structure and function and the content of their extracellular matrix. A substance produced by cells of a specific tissue can contain proteins, salts, water, and dissolved macromolecules. Extracellular, if you think like extraterrestrial, we're referring to life on other planets besides our own. Extracellular meaning outside of the cell. So this is a quick summary showing the general characteristics and functions and examples of where they're located for all four tissues. And also where they're derived from embryonically. Epithelial tissue lines every body surface and all body cavities. All organs are lined on the inside and on the outside by epithelial tissue. Actually, let me introduce some words here. Just a second. So epi means on top. On top. Anytime that you see the word thelium on the end of a word, it's referring to an epithelial tissue. And if you think epi meaning on top, so these are things that line everything. They line all the organs, they line all the cavities. So epi meaning the outer lining, outer covering, or outer covering. So these epithelium always mean anything that ends in the word thelium. Let's not even put that in there. I'm going to put in anything that ends, unless I find an exception to it. Anything that ends in IUM is going to be an epithelial tissue. So epi meaning the outer covering or outer lining, can be on the inner or let's not even refer to it as outer covering. Let's say, actually, no. So epi means on top or outer covering or a lining, some sort of lining. Lining. Some sort of lining. Anything that ends in IUM is an epithelial tissue. I'm making sure, like I'm scanning, in my head, I'm scanning all the tissue types. I'm making sure I'm right about the fact that IUM, anytime you see that, it's an epithelial tissue. So organs are lined on the outside and the inside by epithelial tissue. The majority of glands are derived from epithelial tissue. Epithelial tissue, in comparison to the other tissues, possesses little to no extracellular matrix. And very little outer cellular material within this tissue. All epithelial share several characteristics. Cellularity. It's composed almost entirely of cells with little extracellular matrix. The cells are bound together by several types of intercellular junctions, so connections between the cells. These cells are also polar, and we talked about what polar means by a chemical molecule. Can someone tell me, what does polar mean when we talk about a chemical molecule? Someone tell me. What does it mean to say that a molecule is polar?
It's like when one of the ends of the molecule is positive and the other one is negative.
Right. So it's got two opposing poles. It's got a dipole in it. Epithelial cells are the same way. They have a top surface and they have a bottom or a basal surface. So they have an apical surface, top surface. They have a basal surface, bottom surface. I like to think as basal as like a basement membrane or the basement of a house, where they attach to the underlining cells. So cellularity, lots of cells, little extracellular matrix. Polarity, they have a bottom and a top surface. We'll talk, they also have about the fact that they also have a lateral surfaces. They are attached to things. The basal membrane, remember this is the bottom surface, is attached to a thin basement membrane. So basal surface attached to a basement membrane, which is an acellular. What does A mean whenever it's in front of a word? What does A mean whenever it's in front of a word.
Without?
It means without. Right, that's right, Rachel. So anytime you see a in front of a word, that means without or the absence of. Right, as I write in these words on the whiteboard, write them down. Because we'll see this a lot as we go through the semester. So acellular structure, so a structure that doesn't contain cells, produced by both the epithelial cell and underlying, so tissue that's underneath the epithelial cells, that's made of connective tissue. Also, this is avascular. What does that mean?
It has no blood vessels in it.
It has no blood vessels. So the cells receive their nutrients by diffusion from the underlining tissues in the connective tissue, underlining connective tissue, through the basement membrane. Also, epithelial tissue is highly innervated. It's got a lot of nerves in it to detect changes in the environment at a body or organ region. Has a high regeneration capacity because the apical surface is, remember apical meaning on top. So apical surface is constantly exposed to the environment. The epithelial cells are frequently damaged or die. They're replaced as quickly as they're lost. Your skin is an epithelial tissue. And because the top surface of your skin is always exposed to the extracellular environment, it's constantly being sloughed off and replaced. I don't know what I know they've tried to estimate how many skin cells are lost when we take a shower. It's a lot. And those cells are all dead. All the cells that you lose off the top surface of your skin are dead. They're no longer regenerating. Excuse me, they're no longer actively alive and dividing. They're dead. So we have an apical surface, a top surface to these cells. We have a basal surface to these cells, right here. We have a lateral surface to these cells. We have a basement membrane. And we have blood vessels in the connective tissue. How do you receive nutrients? How do your cells receive nutrients? Somebody tell me, how do your cells receive nutrients?
Food, through sunlight.
Food and sunlight, but how do your cells in particular in your body, because some of your cells are not very close to the surface, how do they receive nutrients?
Through your blood capillaries?
Through your blood capillaries, yes. If a cell is not close enough to a blood vessel to get nutrients and eliminate waste, because that's also how it gets rid of any waste products, is it going to survive?
No.
It's not. This is, if you remember, was it last week? Yeah, last week we talked about diffusion in lab. This all occurs by diffusion. Whether you can absorb nutrients or eliminate waste is diffusion. And diffusion drops off with distance. So the further a cell gets away from a blood vessel, the less likely it is to survive. And if it's not, and if it can't be near a blood vessel, it's not going to divide, it's not going to grow, and it's going to die. Keep that in the back of your mind here. And remember that this tissue is avascular, which means that the only way that it gets nutrients and eliminates waste is through blood vessels in other tissues. So physical functions of epithelial tissue. It's a physical protection from dehydration and abrasion, as well as physical, chemical, and biological agents. Your skin contains as the cells move towards the top surface of your skin, they get this oily substance inside them, and we'll talk about this next week, but they get this oily substance inside them. And this oily substance is hard and it's hydrophobic. What does that mean with regards to water?
It doesn't like water if it's phobic.
It doesn't like water, and it also will prevent water loss. I know a lot of you already work in the healthcare field. What's the number one fear from burns?
Dehydration.
Say it again. I'm sorry.
Dehydration.
It's dehydration. Good job. I thought it would be infection, but it's dehydration because of the oily characteristic of Evan decided to drop a free weight.
No, he's exercising.
He's exercising. I have some free weights here. If you guys heard that bang just now, I have some free weights on my couch here that I use when I work out in our living room. And my one little cat just dropped one on the floor. So number one thing, because skin prevents dehydration. Another way that we know that the epithelial tissue works really well to protect us is in your throat, in your esophagus, you have epithelial tissue. And it's lots and lots and lots and lots and lots of layers. And you want that because if you've ever choked on a Dorito or choked on a chip, you can imagine what would happen if those layers weren't designed to slough off. Second function, selective permeability. So it can regulate the passage of certain molecules in or out of certain regions of the body. Secretion, some epithelial cells are called exocrine cells because they produce secretions such as sweat or oil onto the surface. Sensation, they possess nerve endings that can detect light, taste, sound, smell, and hearing. The basement membrane is a specialized structure of the epithelium, found between the epithelium and the underlying connective tissue. Functions, it provides physical support and anchoring of epithelial tissue. And it acts as a barrier to regulate the passage of large molecules between the connective tissue and the epithelium. We're referring to this stuff right here. Epithelial cells also are strongly bound to each other. Remember, they have these very high membrane intercellular junctions. Several types of these junctions. We have tight junctions, we have adhering junctions, we have desmosomes, and we have gap junctions. So tight junctions, adhering junctions, desmosomes, and gap junctions. Tight junctions work between the lateral surfaces. So they connect the lateral surfaces of each of the cells. Here, right here, right here, right here. And they almost work, if you look at them right here, I would think they kind of work like a thread connecting two pieces of cloth together. So if you wanted to connect two shirts together, a piece of thread that stitches those two shirts together. Adhering junctions, connecting cells together. Desmosomes that have protein plaques, much tighter junction. These are very strong junctions that are full of plaques and protein filaments in the cytoskeleton on both of the cells involved, in both of the cells involved. And then we have gap junctions, which are like little tunnels that are made with connexins. So tight junctions, adhering junctions, desmosomes, and gap junctions. This little one down here, hemidesmosome. Hemi means half. So this is half of a desmosome that connects the basal surface of the cell to the basement membrane. So tight junctions encircle the threads near the cells near their apical surfaces. So they kind of work like a piece of thread stitching things together. Prevent molecules from traveling between the epithelial cells. So keeping things from traveling between the cells and thereby forcing them to go through the cell. Desmosomes, I like to think of desmosomes as being a snap in your clothes. So there's two sides to the snap, and one side fits into the other side. So they appear at locations of mechanical stress between the cells, keeping them from being separated, keeping the cells from being separated when there is a shearing stress on top of them, or when things are moving over the top, keeping the cells together. So tight junctions keep things from moving in between the cells. Desmosomes keep things from breaking apart the cells. And so separating the cells from one another. So their structure, desmosomes, consist of a thickened plaque on each side. So I would think, another way to think of desmosomes is like Velcro. So they have a thickened protein plaque on each side of the opposed cell membranes, with a fine network of proteins spanning the intercellular space between the plaques. On the cytoplasmic side of each of the adjacent cells, there are intermediate filaments from the cytoskeleton that attach to the plaques and provide support and stability. So here we've got the two separate parts. And then we've got parts of the cytoskeleton attached to it. And these desmosomes are the ones that work to resist mechanical stress and shearing of the cells during times of stress. Gap junctions are fluid-filled channels made of connexins. So these are little tunnels. And the subunits for each half of the tunnel. So here's one half of the tunnel, here's the other half of the tunnel. Each half of the tunnel is made of individual connexins. That allow adjacent cells to communicate with each other by the flow of ions and other small molecular molecules through. Are there any questions? Let's take 10 minutes right here and get up and move around.
Okay.
See you back in 10 minutes. I'm going to get up and get a drink of water. Smells really good. Smell really good. Maybe some steak. But I think I'm still slowly learning more and more because I figured out how to find actually. Okay, so let's come back to this. So we talked about the different types of intercellular junctions. Epithelial tissue is classified according to two different criteria: the number of the layers of the cells and the shape of the cells. Simple epithelium is a single layer. So if you think simple, the simplest formation is one single layer, with all of the cells having an apical surface and attached to the basement membrane. Stratified epithelium is two or more layers, where not all of the cells have an apical surface because they're not exposed to the outside, nor do all cells attach to the basement membrane. If you think, anyone in here like to look at rocks and study rock formations?
I don't.
You do? Or you don't?
No, I don't. Mm-mm.
Okay. I've gotten into a little bit of where I've started polishing geodes. I got a geode kit last Christmas, and over the summer and a little bit last spring, I spent some time learning to polish geodes. And when school's out again, I'll get more into it again. But do you know what it means when we talk about rocks being stratified?
No.
So a stratified rock is multiple layers. It looks like it's got layers to it. Bear with me just for a second here. Nice.
So would a
and-
good example of that kind of be like the trunk or the base of a tree, like after you cut it down, how you can see the layers, like the rings?
That's one way, but you know what? I'm going to use that analogy more when we talk about bone. But
Okay.
you're right. That Genesis, that's one really good way of putting it. Let me show you.
Yeah, because I don't really look at rocks, but I've seen a lot of like tree bases or like tree trunks cut.
I understand, and I'm going to be asking you a lot about that here in a moment. And when we start talking about bone, which is in the next 2 weeks. So stratified rock, multiple layers. One layer on top of the another layer. And of course it went away. Really? Give me a second. So 2 or more layers. So stratified epithelium, stratified rock, they have multiple layers. Pseudo-stratified. Pseudo meaning it looks like it, but it's really not. So this looks like pseudo-stratified epithelium, but it's really not, because not all cells will reach, not all cells will reach the apical surface. Not, you'll see that it looks like it could be layered, multi-layered, but it's really not. And we'll in a second, we'll show you specifically how you can tell the differences between the two, whether it's stratified or whether it's pseudo-stratified. So simple, one cell layer. They each have a basal, all the cells have a basal surface, all of them have an apical surface. And all of the cells are attached to the basement membrane. Stratified epithelium, multi-layers. And only the bottom layer are going to attach to the basement membrane. And only the top layer are going to have an apical surface. One thing with regards to this is, where are the blood vessels when you're looking at these kind of tissues? Where are the blood vessels?
Below the basal surface?
They're below the basal surface in the connective tissue that's down here. What that means is that the cells, especially with the stratified epithelium, the cells on the very surface are not getting any nutrients and they're not being able to eliminate waste. This whole layer right here, they're not getting any nutrients and they're not able to eliminate waste. The only cells that are actively dividing are usually this bottom layer, or the bottom 2 or 3 layers, because they're the only ones that are getting the nutrients and eliminating waste. So different layers. So we have simple layers, we have stratified, and we have pseudo-stratified. Epithelial cells are also classified by shape. We have squamous, which are flat, wide, and somewhat irregular in shape. I like to think of squamous as being squished flat, because they look like little pancakes. They're just flat cells. cuboidal cells that are cube-like shaped. The nucleus is usually centrally located, right in the center. Columnar cells, they look like columns, and the nucleus is usually located at the basal region of the cell, the bottom region of the cell. So squished flat squamous cells, cuboidal and columnar. We have simple squamous. We have simple cuboidal. We have simple columnar. We have a simple columnar that's non-ciliated. Cilia means little hair-like shapes, little hair-like structures on the top. So we have simple columnar that has little hair-like structures that's ciliated. And then we have non-ciliated where the simple columnar does not have these little cilia on top. Looks like little hairs. Stratified squamous that's keratinized like that on your skin. We have non-keratinized like that in your esophagus. We have stratified cuboidal, which is 2 or more layers. Let me tell you right now, stratified cuboidal rarely has more than 2 layers. You're not going to see it. If you see 3 or more layers, it's not stratified cuboidal. It can't be because of the diffusion issues with the blood vessels. It's the amount of layers of epithelium is restrained by how close a blood vessel is to it and whether nutrients can be absorbed and whether waste can be eliminated. Simple columnar is never more than 2 layers. Never more than 2. So a lot of times you're going to identify these different tissue types based on a process of elimination. Is it 1 layer? Well, then it's simple. Does it look like it's multiple layers? Okay, then what are the shape of those layers? And how many layers are there? Stratified squamous and what we also call transitional down here have lots and lots of layers. But the stratified squamous is flat on top. Transitional is not, except when it's stretched, but normally it's not. Pseudostratified, meaning it looks like it could be multiple layers, but it's really not. Transitional, if you think, what does it mean to undergo transitions?
To change.
To change. So transitional undergoes changes. It goes from being relaxed to being stretched. And that changes its structure. So simple squamous is a single layer. This would be simple cuboidal. If you look here, these are all really smashed little cells. This is simple squamous. These are pretty smashed little cells, at least in the inner lining. Simple squamous. This is a duct. A lot of our ducts are simple cuboidal or stratified cuboidal, or ducts or glands. Also, underneath and surrounding these, we have a lot of connective tissue. Remember, the epithelium is a lining tissue. So a lot of times it will line connective tissue. It'll line muscle. So simple cuboidal, simple layer, one layer. Non-ciliated simple columnar. You see all the nuclei are kind of at the bottom here, this connective tissue in here. Right here, this is specifically a lot in the intestinal tract. We have a goblet cell that secretes mucus onto the surface of the cell. A brush border of microvilli. Bless you. We have cilia, so ciliated simple columnar. Of the stratified, you can see, actually, never mind. Non-keratinized stratified squamous. You can see we've got lots and lots and lots and lots of layers. Connective tissue down here, basement membrane right here, and then lots and lots of layers. These layers down here are the ones getting the nutrients. These are the layers that are actively dividing, because they get the nutrients and they can eliminate waste. As they actively divide, the cells are moving up towards the apical surface up here. A lot of times, if you want to know what an epithelial tissue looks like, look for an outer surface. Look for a surface and define it based on what you see on the surface. Look for an outer surface on the slide. So apical cells are squamous. The top layer are flat. And you've got lots and lots of layers. So as the cells divide, they move up and they keep moving up. And as they reach the surface, at that point, they're dead. In the case of non-keratin, they don't fill up with keratin. This is what we see lining our esophagus a lot of times. Lining the vagina, we see this. Keratinized stratified squamous, this is your skin. Because it's full, it gets filled with keratin as the cells divide and start to die. They fill up with keratin as they start to die. And if you look here, here's stratified cuboidal. If you remember, I said that you don't normally see more than 2 layers because diffusion of the blood vessels are down in here. Blood vessels are in here. So cells are limited in their growth based on how much blood they can receive, how much nutrients they can receive, and how much waste they can eliminate. So diffusion is what blocks how much growth will actually occur. We'll limit it. So you never see more than 2 or 3 layers of stratified cuboidal, such as the duct of a sweat gland in the skin. Stratified columnar won't see more than 2 layers. Found in the male urethra. The apical, remember, you're always going to look for the apical surface or top surface to be able to define what type of cell epithelial tissue you're looking at on the slide. Pseudostratified. It looks like multiple layers, but it's really not. If you look here, see one layer about right here, you see one layer about right here. See one layer about right here, see one layer about right here. So you see 2 layers. Here you see one about right here, you see maybe one about right here, you see one about right here. We know that this is not stratified columnar. And it has to be pseudostratified because we cannot get 3 layers of stratified columnar. We can only get 2. So this has to be pseudostratified. You look here, we have one here, maybe one layer if we wanted to squint, one layer here, maybe one here, maybe another one here. It has to be pseudostratified because stratified columnar only has 2 layers. That's your distinguishing mark. Transitional epithelium changes shape depending on whether the tissue is stretched or relaxed. So it has kind of this ruffled appearance. And it kind of looks like stratified cuboidal. But stratified cuboidal can only have 2 layers or 3 at the very most, which means that this cannot be it because this looks like it has lots and lots of layers, maybe 5 or 6. Any questions so far? So epithelial tissue also makes glands. Can have individual or unicellular cells or multicellular glands. They perform a secretory function. They produce mucin, which is kind of mucus, hormones, enzymes, and waste products. 2 different types of glands. We have endocrine glands, which secrete directly into the interstitial fluid in between the cells or the bloodstream. Or we have exocrine glands, which have ducts that will secrete onto a surface like the skin or the inside the esophagus. So endocrine into the blood or the interstitial fluid. Exocrine cells, ducts onto the surface of the skin. Via their exocrine glands, via ducts onto the surfaces. Specifically looking at exocrine, we have secretory vesicles. And then we have a duct. And a cross section through it, like right here, looks like this. And this tissue right in here would be connective tissue. A summary of the epithelial tissues. So simple squamous, stratified squamous, one of these is keratinized, one is non-keratinized. I know it's a little blurry. I'm just giving you an overall review slide of the basic types. So simple cuboidal, stratified cuboidal, simple columnar, stratified columnar. I think that's a lousy idea drawing of stratified columnar. And especially stratified columnar, because you're not going to see that many layers. I don't think that's really correct on a very good picture. Pseudostratified, transitional, and glandular. So one thing that you notice here is our glands tend to be simple layers, most often simple cuboidal or simple columnar. Let's not even say most often, let's say always simple cuboidal or simple columnar for exocrine glands. Any questions?
No.
So connective tissue. The most diverse, abundant, and widely distributed and structurally varied of all four tissue types. Connective tissue is the glue and filler of the body. Most examples include tendons, ligaments, fat, bone, cartilage, and blood. Characteristics of connective tissue. Different cells for different types of connective tissue. So different cell types are fibroblasts, osteocytes, and adipocytes. Different protein fibers are elastic fibers, collagen fibers, and reticular fibers. A major ground substance. So a mixture of proteins, carbohydrates with variable amounts of salt and water. Protein fibers and this ground substance, so both the protein fibers and the ground substance are what are going to make up the extracellular matrix. So we have a lot of ground substance. We have cells that have nuclei in them. Cells that have nuclei. A lot of fibers. With blood vessels, a lot of blood vessels in connective tissue. Some connective tissue won't have blood vessels. Lots of fibers. These fibers are collagen fibers. Reticular fibers are the really thin little fibers. The little thicker fibers are the elastic fibers. So the thickest, the collagen, elastic, and then reticular, meaning the thinnest. Major functions of connective tissue. Physical protection. Serves as a support and a structural framework. The binding of structures. Works as storage and transport and immune function. Connective tissue arises from the mesoderm. Two types of embryonic connective tissue. So all connective tissue arises from the mesoderm, which is an embryonic layer, meaning the middle layer. Get 3 different layers formed with a developing embryo. The middle layer is the mesoderm, from which all connective tissue arises. Initially, all of the connective tissue, it comes from mesoderm, and before it differentiates into different types of connective tissue, it becomes embryonic connective tissue, which is called mesenchyme. So mesoderm, mesenchyme, and from the mesenchyme comes all of the other connective tissues. That's where they all differentiate to have their own different function. And then we have the mucus, which is found in the umbilical cord. So 2 types. In mesenchyme, we have mesenchymal cells and lots of ground substance. Few cells and lots of ground substance. To me, looking at this slide, it looks like the mesenchymal cells in the umbilical cord mucus. This is the mucus, this is the mesenchyme. The mesenchymal cells in the mucus look like they're a little bigger. They look like they're a little bit, they're a little bigger than these cells are over here. So the mesenchymal cells in the mucus of the umbilical cord look a little bigger. After birth, the mesenchyme can be classified into 3 broad categories. We have connective tissue proper, we have supporting connective tissue, and we have fluid connective tissue. Connective tissue proper can become loose connective tissue or dense connective tissue. The loose is, we have areolar, adipose, and reticular tissue. In the dense connective tissue, we have regular, irregular, and elastic. And we'll see that this regular and irregular has to do with regular patterns in it versus not having a regular pattern and then being irregular. In supporting connective tissue, we have cartilage and bone. Of the cartilage, we have hyaline and fibrocartilage and elastic cartilage. And in the bone, we have spongy and compact. In the blood, we have fluid connective tissue. Or the only fluid connective tissue we have is blood. So connective tissue proper, loose connective tissue, and dense connective tissue. In the supporting connective tissues, we have bone and cartilage. In the fluid connective tissue, the only one is blood. And they all arise from the mesenchyme, which arises from the mesoderm in the three germ layers of a developing embryo. In connective tissue proper, so referring to this little section right here, in connective tissue proper, there are two groups of cells in present. There are resident cells and wandering cells. Resident cells include fibroblasts, adipocytes, macrophages, and mesenchymal cells. In the wandering cells, we have mast cells, plasma cells, free macrophages, meaning they can wander, and leukocytes. Do you remember the word phagocytosis?
Yes,
What does it mean?
it meant that the cell was eating something else.
It was eating something else. Right, good job. So when it's eating a phage, anything that has the N word of it here is phage, it's a cell that's involved in phagocytosis. So macrophages engulf other cells, or engulf bacteria, or engulf debris. Fibroblasts produce fibers, so they produce our elastic, our reticular, and our collagen fibers, and the ground substance of extracellular matrix. Adipocytes serve mainly as lipid reserves. These are our fat cells. Fixed macrophages phagocytize foreign materials. Mesenchymal cells divide in response to injury to produce new connective tissues. Mast cells will release granules such as histamine and heparin to stimulate local inflammation. Histamine, if you struggle with allergies, I struggle with allergies. Struggle with allergies, histamine is your major culprit. That's why most of your allergy pills are antihistamines. So you're mainly, a lot of what you're looking at when it comes to an allergy pill is trying to block these mast cells or neutralizing what they release.
So is the word heparin referring to like heparin we use for blood thinners?
Exactly, that's exactly what it is.
Okay, I didn't want to correlate it and be incorrect.
No, you're right. You're exactly right. Using a blood thinner means that you will stimulate blood flow to that area versus having it get clotted up. And especially with local inflammation, we want to increase the permeability, the blood permeability in that area rather than reduce it. Plasma cells form antibodies that bind to foreign substances. Free macrophages, phagocytes, leukocytes attack foreign materials and combat bacteria. Bear with me just for a second here. I'm going to put something up here. I'm gonna shift gears again. So just a little bit of a summary of that slide. And just some basic words to remember. Anytime that you see on the end of a word site, that is a cell. It means cell. And this is sometimes a cell, this is a cell that is not actively dividing or secreting substances. Actively dividing or secreting substances. Of course, I went into caps lock. A cell that ends in the word phage, like Miranda said, is a cell that can perform, is involved in phagocytosis. And if you think cytosis meaning a cell, so eating other cells, eating other bacteria, eating other cellular debris. So cytosis meaning other cell debris. Blast is a cell that's building, usually by secreting matrix. So building, actively building or secreting something, creating, let's say matrix, just to make sure. This cell, not actively dividing or secreting substance. This site cell may also be in a state of almost senescence. So a lot of times, if you can learn that what the beginning of the word is, like for a adipocyte, adipo means fat or lipids. So adipocyte has to be a fat cell. A blast cell is an actively building cell, like a fibroblast. You may not know what I would think fibroblast, if you think fibro could mean fibers. Fibroblast. Just based on the word alone. Cell that secretes fibers. Remember, that's the part of the ground substance, fibers, and maybe other things. So there's three major fibers produced by connective tissue cells that are secreted into the extracellular matrix. We have, oh, wait. We have collagen fibers. Collagen fibers are long, unbranching, strong, and flexible fibers that are resistant to stretching. They make up approximately 25% of all protein in the human body, making collagen the most abundant protein. We have elastic and reticular fibers. These are thinner than collagen. They stretch easily, they branch, and they rejoin. These fibers allow structures such as blood vessels to stretch and relax. Reticular fibers are thinner than collagen fibers and form branching networks of a woven framework. These are found often in the stroma, so internal areas of an organ, with abundant spaces such as the liver, lymph nodes, and spleen. These are reticular fibers form a filtering network. So we find these in organs where we find a lot of filtering capabilities, like the liver, like the lymph nodes, like the spleen. Elastic fibers allow things to stretch. Collagen keeps things strong and flexible and resists stretching. If you have heard of them, bear with me just for a second because I have to remember what it's called. For some reason, I can never remember what this disease is called every time I think of it. Marfan syndrome. Marfan syndrome is what they think Abraham Lincoln had. Usually very, very tall people, very, very narrow faces, very thin hands and feet with very long fingers and toes. If you've heard of basketball players who are running up and down the court in the middle of a game and collapse and die from a heart attack, oftentimes it's a connective tissue disorder that's attacked the collagen fibers, and it's a defect of the collagen fibers. And so this is what they call Marfan syndrome. Bear with me, I'll type it. Stop sharing one more time and I'll type it in here. Marfan syndrome. So I'm gonna look and make sure that I'm right about collagen defects. Of course, it doesn't tell me that, you know, this is, and one more sec, I'm gonna look one more place. Mom? Yeah. You know if Marfan syndrome, it's can you back me up and say that it's a collagen defect?
Say again,
Collagen defect,
what about it?
Marfan syndrome.
I don't know. No, I can't. I don't know.
Bear with me, guys. I'm looking it up because I don't want to tell you the wrong thing. Honestly. Okay, give me one more. I'm gonna look one more place, so then I'm gonna give up for a moment. Oh, it's not that bad. Genetic. Right now, go with the fact that it's a collagen defect because I'm almost sure of it. I can't remember exactly, but almost every semester I talk about it and I know that it's a collagen defect. So if you think about that, the reason Marfan syndrome kills people is because it's a weakness in the collagen, which is the most abundant protein in the body, that should be resistant to stretching and should be strong and flexible. Well, what about if it's not? That's Marfan syndrome. So ground substance, coming back off my tangent there, ground substance of connective tissue is a combination. So we have fibers, the reticular, the elastic, and the collagen. Then the rest of the ground substance is specifically proteins and carbohydrates. And the texture is usually gelatinous, so like a gel. It can range from being like a semi-fluid, like adipose tissue, to being hard like bone. The loose connective tissue serves as the body's packing material found in spaces around organs. Types include areolar, adipose and reticular tissue, dense connective tissue, like dense regular and dense irregular and elastic tissue. Areolar connective tissue tends to bind and pack around organs, surrounds nerves, vessels, and the subcutaneous layer, subcutaneous meaning under the skin. Adipose connective tissue, mainly adipocytes. And it's also found in the subcutaneous layer underneath the skin, surrounding the kidney and others around other selected organs. Reticular connective tissue, found in the spleen, the bone marrow, the lymph nodes, and the liver, because these are areas where a lot of filtering occurs. Or the lymphatic system filtering occurs. Dense regular connective tissue, tendons and ligaments. Dense irregular connective tissue the dermis capsules of organs. Elastic connective tissue in the walls of the large arteries. It's also found in your ear, allows you to flip your ear. So areolar connective tissue contains fibroblasts. Remember, these are the ones that secrete fibers. Fibroblasts and collagen. I like to think of areolar tissue. It's a very loose tissue, has a lot of air spaces in it. I think of it as being a very airy tissue. Very loose tissue with fibroblasts in it. Specifically collagen fibers and elastic fibers. Adipose tissue contains adipocytes. Remember, these are cells that make up adipose or fat tissue. You can sometimes see, a lot of times you will see that you'll see multiple tissue types of connective tissue or multiple tissues in one slide. So adipose tend to see everything squished up to the side because the inside is filled with lipid or oils. And all the nucleus of the adipocyte and the extracellular area of the cell is squished up to the side. The cytoplasm is all squished to the side. Blood vessels in between. Reticular connective tissue looks a lot more like a filtering screen. Reticular fibers and leukocytes. So remember, the one thing that we haven't really talked about, leuko has a lot to do with the lymphatic system. So in reticular tissue, you find a lot of leukocytes because reticular connective tissue is found in the lymph system. Some fibroblasts and some leukocytes. Dense regular connective tissue. The collagen fibers. So we've lots of collagen fibers here. And they're lined parallel to an applied force. So like all of the connective tissues we've talked about so far, you see a lot of fibroblasts. But in dense regular connective tissue, the collagen fibers they have a pattern to them. All of this, here's the cells. You can tell based on the nuclei that are staining purple here or black here. All of this here is ground substance that's being secreted by the fibroblasts. And the ground substance is substance and collagen fibers. Dense irregular connective tissue, you have fibroblasts, but then the fibers don't have a regular pattern to them. That's why it's the irregular connective tissue. It's pretty tight, it's very dense, tightly compacted. But there's not a regular pattern to the fibers. See that here, it's very tightly compacted. If you compare this up against this tissue or this tissue or this areolar tissue, very tightly compacted. One tends to have a pattern to it, the dense regular. One doesn't have such a pattern to it, the dense irregular. Dense regular, dense irregular. Any questions?
No.
I'm following along on the PowerPoint on my own screen, but we can't see the PowerPoint on the screen.
I can't hear what you're saying. Can you say that again?
Sorry, I'm following along with the PowerPoint on my screen, but we can't actually see the PowerPoint on your screen.
You can't see, you're not seeing the PowerPoint, my PowerPoint?
Nope.
No, not.
Okay.
Maybe.
Yeah, feel free to stop me when you're not seeing it, guys. I don't know what slide this is. Bear with me just for a second here. This is slide 59. So elastic connective tissue, elastic fibers have the ability to stretch and recoil. Just a summary slide here. Loose connective tissue like areolar connective tissue. Dense, say it again. Dense irregular connective tissue. Dense regular connective tissue. Elastic connective tissue. Adipose and reticular connective tissue. You want to learn to recognize one from the others, be able to identify each type. The best way to do that is to Google images, because there's lots of histology images out there. Google image specifically looking at adipose tissue. Google for images specifically looking at reticular connective tissue. And the more and more you look at it, the more and more you're going to see specific patterns and be able to recognize one from all the others and recognize each one. Supporting connective tissue, bone and cartilage. Cartilage is a firm gel-like extracellular matrix composed of proteins and ground substance. In this case, the cells are called chondrocytes. Anytime you see the word chondro, that means cartilage. So these are chondrocytes. Chondrocytes occupy small spaces by their enclosed by their extracellular matrix called lacunae. Lacunae are cavities. So they reside in little cavities. And the cavities are surrounded by the extracellular matrix that the chondrocytes of chondroblasts have produced. If you remember, blast is a secreting cell or building cell. So cartilage cells support and withstand deformation. Three basic types of cartilage. We have hyaline cartilage, fibrocartilage, and elastic cartilage. Hyaline is the most common type, but it's also the weakest. It has a glassy matrix to it. Hyaline means glassy, got a very glassy surface to it, glassy sheen to it. Here's our little chondrocytes living in these little cavities, these little lacunae.
I know. Over here.
Fibrocartilage has little collagen fibers interwoven in it. You don't see a lot of fibers in hyaline cartilage. You just don't see them because it's more of a glassy sheen to it. But you do see it in fibrocartilage, lots of fibers.
Mm.
And these are collagen fibers. You see, these are your intervertebral discs. This is, where else is this found? Your intervertebral discs, your the cartilage that connects your ribs to your sternum is fibrocartilage. Hyaline cartilage is what you find in your nose. Your bone is only the most inner part of your nose is what's made of bone. Hyaline cartilage is what actually makes up the front structure and the pokey outy part of your nose.
Kitty, 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 kitty.
Elastic cartilage, abundant elastic fibers, lots of fibers. And if you look here, one thing you see is a lot more convolution in the cells in the elastic fibers. There's a lot more of the cells. So a lot less cells in the fibrocartilage, a lot more in the elastic.
You didn't like that, did you?
In bone, osteocytes are the major bone cell. Osteo means bone. So osteocyte is a bone cell. The extracellular matrix is a mixture of collagen and calcium salts. This mixture provides flexibility from the organic components and the compressional strength from the inorganic components. Someone was telling me that they like to look at tree trunks. I think it was Miranda. We see a lot of tree trunks. And in the case of these tree trunks in compact bone, we have these rings or circles that look like sections or a tree trunk are called osteons. And each of the rings are called lamellae. Each of the rings are called lamellae. The osteocytes are the little black parts along the rings that reside in the lacunae. So the whole thing is a lacunae, and the osteocyte lives in it. And then the central canal is where the blood, the vein and the artery and the nerve run through the bone. The central canal is also called the Halversian canal. Any questions? That's one. So fluid connective tissue. This is blood. So composed of the following components are plasma, a watery ground substance containing protein fibers, erythrocytes, we're seeing that sites again. Urethra means red. So erythrocytes are red blood cells. Leukocytes, which are part of the lymphatic system, are called white blood cells. And platelets are fragments of blood cells that are involved in blood clotting. The formed elements of the blood are everything except for the plasma. Formed elements meaning they have a plasma membrane surrounding them. You know, like, yeah, they have a plasma membrane surrounding them. So the fluid connective tissue or blood contains erythrocytes, leukocytes, and platelets. Plasma contains soluble proteins. If you look here at all of the red blood cells, they look like little donuts. What their structure, they're like a donut shaped cell, but they do have a middle. They kind of look like a flattened basketball that's been punched in on both sides. And so the center is very, very thin to the point of where it looks clear through a microscope. The little tiny pieces are the platelets. And the bigger cells that you can see a nuclei in them are leukocytes or white blood cells. And then a summary of the connective tissues. Are there any questions?
No.
No.
So just to summarize really quickly here. Epithelial tissue. We have squamous, we have cuboidal, and we have columnar. We have simple squamous. We have simple cuboidal, and we have simple columnar. We have stratified squamous, which when you're looking at a slide, which you'll see some slides in the activities for this lab. If you look for the outer surface, that's how you're going to know that it's stratified squamous, because it's going to look flat on top. If it's stratified cuboidal or stratified columnar, it's not going to have more than 2 layers. If it looks like it has more than 2 layers and it looks kind of ruffled on the surface, got kind of a ruffly pattern, that's transitional. If it let's see here. If it looks like it could be a combination of stratified columnar and stratified cuboidal, and it has more than 3 layers, it's pseudostratified. Of the connective tissues, we have connective tissue proper, which includes areolar, adipose, reticular, dense regular, dense irregular. Then we have supporting, which is the cartilage, and we have hyaline cartilage, elastic cartilage, and fibrocartilage, and bone, which is also called the osseous tissue. Of the fluid, we only have one, that's the blood. And we rarely end before 12:10, so let's count this a win. We have 3 minutes. Do you have any questions? I will see you next week on Tuesday.
Thank you, Professor.
Thank
You're
you. Have a
welcome.
good one.
You too.
Have a good one.
You too.
Oh, I have a question.
Okay.
So I know this is definitely on my part.
Okay.
So I did my lab quest, but I completely missed my lab, basically lab, like the one that's due every Wednesday.
Is it still open? Is this Asante?
No, this is Michaela.
Michaela. All right. Let me see here.
I will put my camera on, but I have my bonnet on.
Don't worry, you're fine. Give me 1 second. I'm gonna try to fix it right now.
Okay.
Give me 1 moment. How do I do this? Let's see. I learned how to do it last night, so be patient with me for 1 moment.
No, that's okay. Take your time.
Kayla, costly is your last name. Okay.
Yes.
All right. I've got it set up so it will be next on the 29th at midnight.
Okay, thank you so much. I know that was definitely my fault. I was doing my one assignment. I was like, I feel like I'm missing something, but I didn't see it on the due, like where it says things are due. So I was like, well, maybe I'm not. Maybe I got on my work end.
I understand.
Sorry about that.
You're fine.
Thank you.
You're welcome.
Have a good day.
You too.
Bye-bye.
Jessica and Stacy, are you here?